Hello, everyone. Welcome to this week's Quant Seminar. And our first speaker today is Jianhua Xing. Professor Xing received his bachelor from Peking University, master from University of Minnesota, and PhD from UC Berkeley. After being a postdoc at the UC Berkeley and an independent fellow at Lawrence Livermore National Lab, Professor Xing went to Virginia Tech as a faculty. He then joined the University of Pittsburgh in 2015. He's now a professor in the computational and system biology department, the School of Medicine at the University of Pittsburgh. His research uses statistical and chemical physics, dynamical systems theory, mathematical and computational modeling in combination with quantitative measurements to study the dynamics and mechanics of biological processes. So without further ado, let's welcome Professor Xing. Professor, you may want to start.
Yes, thank you so much. Thank you very much. By the way, before that, I want to ask how long should be? I thought it's
1 hour.
Okay, good. That's what I thought. Okay, just want to make sure. Yeah. So everyone, I'm very excited to very honored to talk in this audience. Because I read George and other guys' review. I'm really impressed. That's she's very relevant. So then George asked me to give a talk. So I want just discuss some of our recent efforts, which really you will see are very align what you guys doing here. So this is reconstructing cellular dynamics from single cell data. So first, let me just talk about the biology question. The biology question is this: in our body, there are many different cell types as shown here. Now you can tell, right? Even not with being a biologist, we know there are a lot of they're very different, but we also know they come from the same fertilized egg, right? Have the same set of genome. And also they can change phenotypes. So that's what happens during developmental process. So the embryo, you see the cells, they can develop into different cell types. Specifically here show one called epithelial to mesenchymal transition. It's an important process in development, and also unfortunately hijacked by cancer metastasis things. Then another example is reprogramming. So we can people can reprogram terminally differentiate the cells into induced pluripotent stem cells. So biologists, biomedical researchers are very interested in those things because they want to know how do cells regulate phenotypes and how to control the cell phenotypes. And then the biologists develop a lot of tools. So they use CRISPR, imaging, all the things. But the question I want to ask is, okay, so this is the language biologists talk about. But in physics and engineering, right? The language we talk about is very different. I've shown here is a pinball machine. So we know how we do that. Well, okay, here's a system. I can write down a set of ordinary differential equations, for example, like this, the Newton's equation. Then I can use the equation to describe how the system will evolve over time. And we know this is in the history of physics that happens really starting from very early time, like Chopin's Tickle. So he collects a lot of data, right? Planning motion things. Then his student Kepler, he did this. You will call this data mining, right? So this data try to mine some kind of rules. But at the end, we got this mechanistic theory. That's this equation. That's how we can describe those planet motions and motion like a pinball, and also like even shown here is COVID-2 virus protein. We can simulate that. So this is how we develop in physical sciences. So then the question is, can we do similar thing for, again, we now have data-driven, right? Data rich. Okay, so beside this data mining, we can go to mechanistic dynamic theory. And that's where, if you think about our system as a dynamical system, so we should be able to write down a set of equations like this, very generic, about that shown here. So this A really tells the Z is something of the dynamic variable. We need to identify, right? There's one more task was to identify the proper variable, right? Then there are relations, so we can reconstruct the equation. So this is the task I try to ask. And from mathematical perspective, let's look at how you generate, let's forget about the Z first. Let's just see how we can get this A. So mathematically, you have say the system set of describe a generic set of equations. Then this is a Z, as we know it's a state variable, right? We assume this can completely specify the internal cellular state. So that's why we say this is we use this Markovian dynamic, right? Zeta is a colorless noise. Let's put this way. So you can write down similar partial differential equations, and here you talk about convection and their diffusion, and you may consider there are some cell death proliferation that can describe by this term. So N is the number density. So then you know this J is the flux term. So this is if you easily can link to what this A is, especially under Stratinovich is very simple, just directly to A. If you Ito, you have actual term. So this is the mathematics behind this. Then you see this immediately suggests how we can use data to help us do this. One thing is you measure your system, the distribution density, number density along time, then you can do regression. So what clearly doing this, there are several work they use this, even use optimal transport of things. So a problem of this, we were talking about very high dimensional density function to measure. And so it's very sensitive to the accuracy of your measurement. So another thing, another way to do it is suppose you can measure both Z and also it's the instant velocity. So then we can see, okay, so we can use this. So in practice, you relax your data by some bins, right? So you can get this. So this is kind of a mathematical basis for our discussion. So with this, let's talk about this cellular system. So the cellular system I'm talking about here is, let's say there's a cellular process we're interested in, and we want to write down a set of equations to describe it. This has been done in mathematical biology or system biology to talk about. So here I show you generically you have two genes, and there what people usually do is, okay, there are a lot of experimental data. So we collect all the data, and the biology tell us, okay, there are two genes important, and say there's gene 1, X1, that have self-activation. Then as a modeler, we do is we'll just translate this knowledge into a mathematical equation term. So like say this is the dxdt, the x1 dt is we describe the x1 gene expression, how to change, right? So this is the transcription of x1, and this term reflects this is self-activation. Got this function form, you see, this is an increase of x1, and then there are x2 people. Oh, there's also self-activation. So we write down a similar thing. There are some parameters. Then there's mutual inhibition, where you write down another form. Then it's this one, see this inhibition. Then you have degradation. So you got these mathematical equations. There are some parameters. Use the data. Try to learn, constrain those parameters. Then you got the set of model. Your model. So you got now you have a math model. You can do analysis. So we mathematically now we see, okay, this is x1, x2 give you the state space, right, phase space. So dx dt give us the vector field. So for any given cell state, we have a vector that tell us where to go and how fast it goes, right? So then we can do a lot of analysis, like a bifurcation analysis of things. So I'm shown here, you can see there are several different attractors corresponding to different cell types. Okay, so this has been done for decades. But the problem is, you see, even for this small system, you need a lot of information, you need the data to constrain. But we know in real biology, it's not just small networks, it's many highly connected, interconnected network. So can you reconstruct genome-wide vector field? And then you need, well, see, this is impossible because you need a lot of data. Well, then, so today I want to just tell about a paper we published recently. So this is mainly done by postdoc, two very talented young researchers. One is Xiaojie Qiu from Jonathan Weissman Lab at MIT, and Yan Zhang is my student. So They and collaborate with many other colleagues, as shown here. So we developed this work. The ideas come from here. It's based on a paper published several years ago. If you have known this field called a single cell RSeq, so what people did is you have a cell, right? You have a cell, you just destroy the cell, then you can measure. Called this, give some biology, right? The gene, we talk about the genes, right? The gene will transcribe into mRNA. So the mRNA can be used as template, translate into protein. Protein has a lot of functions. So in this single cell RSeq one, people essentially to measure the number of mRNAs for each gene. So then you see you got a vector, right? You got a vector in this. So each gene has the gene copy number. So then you use this vector of gene copy numbers to represent the cell state. But you see the problem that you have destroyed cell. You destroy cell, then you cannot get the dynamics. So this is kind of a dilemma, right? But those cells, okay, those people say, okay, actually you got some dynamic information. So their argument is this. So this is the gene, you do transcription. Your transcription got mRNA for the unspliced mRNA. So means these actual things, you see. Then they have to remove those actual things, got the spliced mRNA. So this become mature mRNA can use that template for translation. Then eventually degraded. So you can
Jianhua,
yes.
What is S? So alpha S is some state that you describe, but what is S? Why you have the dynamics in terms of S? Is the coordinates?
Yes. I'll just talk about it. Exactly. So I said that, to represent cell state, we have this vector, right? So here, what is S? S is I just show here that you see we have unspliced mRNA, unspliced mRNA. Those two things you can read from your data because the data, one thing from the you found this intron called intron. This one do not have intron, so you can distinguish U and S. All those come from your data. So S is a spliced mRNA. This is unspliced mRNA. For a given gene, you can count, okay, in one cell, how many copies of the unspliced mRNA, how many copies of the spliced mRNA. So that's the data, right? And what I talk about from dynamic perspective is, okay, for each step, you can write down a rate equation. So I have this alpha, I just mentioned beta, gamma. This is a transcription rate, splicing rate. Gamma is the degradation rate, right? But you may say, why I put the gamma alpha S here? This is exactly what I'll talk about it. But first, before this, let me finish this one. So you see, we from the data, we have U, we have S. This is from data. Gamma is something, the degradation constant, you can estimate some way, you find a way to estimate it. Then you got this, you have U, gamma, S. So you can define this called R velocity, you can the instant velocity. It's estimation. So this is, then you can give something about it. So what we look at, we say, okay, this is, we say alpha is really not a constant, because what alpha is, this gene, this is talk about transcription. For gene transcription, it's regulated by many factors. They are called the transcription factors. They can come to bind the promoter region, then that will make this transcription to be faster or slower, right? So this is alpha's function. So why I put alpha S? Well, in the real system, you have this mRNA to go to protein. Protein is where affect is transcription. But since we can only measure this mRNA, so here let's mathematically assume, first of all, we say it's S is enough for us to specify the cell state. Although you in reality you have to have the proteins that then we say the other thing, right? So here with S is complete, then become U will be also a function of S, right? So then also assume this Markovian dynamic means my alpha, this transcription is a function of my current S, although in reality some time delay, right? So we have this two assumptions. Then you realize this is eventually is a function of S, is a complex function, but this function contains information of the gene regulation, and this function F is what I talk about here. Okay. So now you see the question is, now I have all the data. lot of data, lot of copies of a single cell data, right? Discrete. Can I learn this F function? But this F function is a nonlinear, multidimensional vector function. So how can we learn this? So that's our task. Well, the first thing is that we want your input to be more reliable. So the first, the paper I just mentioned here, it's have a good estimation of this R velocity, but there are still a lot of problems. Important one thing I want to point out is this gamma, actually, I want to say this should be gamma over beta, right? Then people just said beta equal 1. So all this relative. That there are many other things. So then there's something people use, they can directly measure the MR tone dynamics, use some, the techniques we don't need to worry about. And then, what means that they essentially, they can use certain things to label, can distinguish this nascent and the mature MRA, so the old MRA. So from there, that's a machine learning process, which I think is very standard. So I won't go too into detail of that step. But from a lot of data, you try to learn those parameters. Essentially, a lot of regression method type thing. We discussed here with different method, different type of data, how you try to infer this alpha, beta, gamma. These parameters, and this is show some results. Here, indeed, for this set of we show, okay, this is a human cell line, this is a mouse cell line. And
Jianhua,
yes.
because we are interested in methods, can you say more, please? For example,
yeah,
you seem to have these Bayesian models are what kind of hierarchical Bayesian? What kind of modeling are you doing here? I see some deterministic and some stochastic and.
Yes, that's good. So I
we were interested mostly in methods. So
I know. I will actually I will talk about some because I thought those are kind of standard, so I didn't go to. So what we do is like I shown here. We have a lot of single cell data, right? Single cell, they have their expression. Then we try to learn this parameter. You see, there are a lot of parameters. So there's certain, let's say this is regression methods. So assuming this is stochastic as a deterministic ODE, then we try to essentially fit parameters. So then we do have there's stochastic. Then you have this moment-based method. But sorry, I didn't put the slide on this because I thought this is relatively straightforward methods. I didn't put, but I want to really talk about some other methods. Other parts. Well, I'll go into detail, and as I mentioned, I'll skip those. Skip them the biology part. Okay. So now to the second step is you have your discrete data. You want to learn this function f. Since we don't know the function, what its function is, so that means we talk about a function space. We try to find what's the best function to fit your data, right? That's put in a simple way. You do is we work in this Hilbert space. Well, it means you have a basis function, you expand it, so you can do you try to find what's the best coefficient. And this is called the reproducing kernel Hilbert space. So you see, essentially, you work in both in the function space. So it means you have this basic function, then you try to determine C, or in the vector space, so where the data space. The data space in this duality, in there you see C is vector, C vector, and then you try to then you for each cell, you have a data point, you have a given point. So you want to find what is a vector. So then you see this is you have essentially you have several points, reference points. You have their vector, and then you have weighted by this. So you see this is a duality, right? Depends on which space you talk about. So one is the coefficient, the other basis. So this is the reproducing kernel Hilbert space, which I think is familiar to this whole field. I want to go into detail. This is mainly to explain to those biologists what this means. So basic. But you means we work on this basic function. So want to then the learning step is we try to, step is we need to define a loss function. The loss function that help us to determine what's its coefficients. So this is the loss function. The loss function is this, right? So, so this f is my calculate. So given the data point, this is the data PVI. It's a measured velocity, and it's also x is a state, right? So there we can from this we can get this f from calculate, based on a given set of a coefficient calculate f. Then we try to minimize. Then we try to minimize the distance between the measured and your calculated. So pi is the weight. If you want to put some data to more weight, so that's give you that. Then do you have this regularization? So you want to minimize the modulus to avoid overfitting. And so the f, as I just mentioned, is a linear combination of the basis function. So here the basis function is the Gaussian basis function. So it's shown here is so we have several, we have number of in this high dimensional state space, right? It's a high dimensional state space. So we have some reference points. So those fixed reference points. Then for any specific point x, that's so this is its weight will influenced by this weight by this reference point to be decay in the Gaussian way, right? So that's the basis function. Then with that, since this we define an inner product. Inner product, then you got this. This is the norm. So so then your loss function can be written in this form. Okay. So the procedure now becomes you have your data. So this discrete data. Then this is your function you want to fit, optimize, right? So you try to minimize this loss function. So you do you will learn, eventually you'll determine this C, you got this function form, it's vector form, function form. So with those process, you will be able to get this thing is analytical form of function form, right? We already have, I show you the Gaussian, the exponential, the Gaussian basis function of things. So you got the analytical function, then you can do a lot of other things. Then I'll just mention again.
Yes. Sorry, can I interrupt? Can you go back to the, here, I have a question. So this X tilde, is it is known or?
Yeah,
oh, no.
X tilde, yes. X tilde is, it's some, you preset some reference points, help to generate a basis function. So the more data you choose this, you have more basis, right? You want to have a sufficient basis function help you to represent this function form.
Yeah, so for X tilde, we don't need observations over.
No. X tilde is fixed.
Yeah, is there any?
It's a parameter.
I know. Is there any technique how to choose the X tilde?
We just essentially from our data set we randomly choose a number of it, and then you can find out if you have sufficient X tilde number, then this is result pretty much very stable converge.
Okay. Yeah, thank you.
I'm not sure there's any other way to do that. We use Latin cube, lattice, other things maybe. We didn't explore that, so this is something can be. You want your X tilde to be kind of cover.
Yeah.
The space.
Yeah, of course. Yeah, thanks.
Hello, this is Lulu. I also have one question for the method. So what's the advantage of choosing this Gaussian kernel instead of, for example, can we apply just a neural network here?
Good. We it's just simply for convenience. For this one, we haven't explored other basis function will give better one, but we are thinking about, because this is for convenience. Gaussian have a lot of convenience, mathematical convenience, but I agree this mathematical biologically interpretation is a problem. So we are exploring if in another way, but the first thing is, okay, this is the convenient one, see if it works. That's where we got. Yes,
Okay,
but this is definitely a question one should ask. Okay, so you got those. Then I think I'll quickly skim this. So you got some data. You show, oh yeah, it's good result. We got a lot of things. But one thing is important is that I want to tell you. So in this field, people, a lot of informatics people, get this statistics. But I'm as a dynamics background people, so I'm interested in dynamic equations. So there, you got this. I don't know if you guys are familiar with the molecular dynamic simulations. So I just shown, right, the thing. So you got that force field. Here's vector field, right? The force field, then you can do a lot of simulation, do a lot of things. Here, essentially similar thing. You got the force field, you can run a lot of simulation, that's shown here. And you have vector fields, you can do some analysis. Here, so you do this Jacobian analysis, because Jacobian give us the information of the gene regulation. It's activations. So biologists need to do a lot of very carefully designed experiments to infer this. But now, with single set of data, you got this, you can immediately get it from this. So I show here this. There are two genes, very important, and people in decades of study, they already established the two genes. They have mutual inhibition, self-activation, and we show here. You get from the data, and this is because active shows their inhibition, active self-activation, and then you got the quantitative information. So it means that okay, if you increase the amount of one, the regulator, how that changed the effector, the gene transcription. So there we show, okay, you got this. So if you remember in my talk, so that's dose response, right? This is called a Hill function, sigmoidal, means how the effect is. So we got this, and show here. This is this is see there are two genes, right? They one act on the other one and reverse. They are not symmetric. And then we found out in 20 years ago, those biologists, they have done experiment. They have done very, I would say, it takes us postdoc a long time to do all those, generate a lot of things, go into human cells and do those things. But now with this single set of data, we got this quantity information, and we not just this two genes, but genome-wide. So I think that's the power for this data-driven learning, machine learning. And you can ask why. And you can also do more analysis, because the vector field, you can do, this is streamlined, right? You can do this like look at this higher order different derivative, and this this is curvature. So all those tell something about gene regulation, because this is where the system start to regulate. So there, I won't go through in detail. Means you will find out where. Well, let's put a simple analogy before I say. You drive your car, right? You drive your car, and then you push your gas. Then the car start to accelerate and speed increase. The moment you press your car, the speed hasn't go up yet, right? But you're pushing it. The cells start to the car start to accelerate. So that's where you actually start to regulate. But the effect can be observed later. So those biologists, they typically observe the effect, means that the speed really goes up, the gene got expressed. But then that's too late. This commitment already taken place a long time ago. But now with this, we can find out where you press your gas. So that's we can show people a lot of things. And then and also with the vector field, you can ask, about a question: if I change from one cell type to another cell type, what's the optimal path? Because mathematically, we know there can be many different paths with different weight. So you can write down this path syndrome type thing. So you can write down, define the action to minimize, to find out what's the optimal path. Then you can ask, predict how the paths go and what are factors. So they can manipulate to change, to regulate the transition. So we predict this and agree with the experiment. Okay, since this is a math work, now I want to really talk about something, some details, and also especially this part I want to seek some comments and some input. So this is also related to, we talk about the physics informed machine learning, right? So means in your system, a lot of prior information help us to do. Here's one thing in the system. We know the system, we assume the system obey these dynamic equations. And then we know mathematically there's constraint. The constraint is, this X, the DXT, and X is the data, right? That gives us, we know a full dynamic system falls into a dynamic manifold, okay? And we know that the DXT should be in the tangent space of this manifold. So that's the constraint, right? So, but this is a mathematical constraint. But thing is, in real data, the real data is noisy, and also we know the DXT as estimate. So there are certain assumptions involved may not be true. So then, in reality, the data is not really unlikely to be in the tangent space. So how can we? So what actually when we do this analysis first, we need to kind of do a projection to remove this orthogonal component to make it is really force it to go tangent space, because here we have some, we have the data X, the X state, the state variable, the measurement is more reliable. So the DXT is derived less variable. So we want to use that as a reference kind of to correct this DXT. So that's how can we do that. And then so you remember when we do this, we usually not in the 2,000, 20,000 gene space. Rather is we do a dimension reduction lower dimension. Because the data, we know the dynamic system is a low dimension manifold. We utilize it. So, but how can you project a vector field into a low dimension, right? You know how to change data, right, to a low dimension manifold. A lot of dimension reduction method. So how about this vector field? And we know the system. I only talk about this F. So we know the system actually is stochastic. How we add the stochastic terms. Then for practical usage, can we make a kind of discrete transition model? That's for some convenient analysis instead of this continuum dynamic equation. To address this question, so here I want to mention, actually in this original paper, they have very clever idea. They don't realize that this tangent space of the arguments, but they do need to project on low dimension manifold. So they suggest a very interesting idea. The idea is, okay, suppose you have a point, right? This is a cell, and then you have a neighbor cells. So you got this neighborhood, and then you assume this dynamic is kind of it's a transition. You can define transition matrix from have probability to transit to my neighbors. So they define, and clearly if my neighbor is this is right, this is me, this is neighbor. So this is a vector, and if the velocity vector is along this distance vector, then we know it's likely this will transit this way, right? If this opposite, then unlikely this cell will go here. To utilize this, you can define this correlation coefficient, essentially their cosine correlation between two vectors. So use that as a matrix to define this arbitrarily, essentially kind of arbitrarily define this transition probability. As shown here, it's normalized, right? Normalized transition probability. So then you can express your velocity vector by this displacement vector. So you see it got weighted by this transition matrix, transition coefficient. Okay, then here this K, well, our K is they want to correct some density, because when you do the measurement, clearly will bias to those you measure you have more sampling, because the sampling is not uniform. So you got the arbitrarily define this. So with this, now you see how you can project low dimension, because no matter which representation you use, if these cells have probability to represent to go this cell, then this should be maintained, right? Then the data you can represent in different representations. So that's how you can, a very simple way to project. You can do this projection to low dimension. To maintain right, these relations, right? But problem here is there's a lot of arbitrariness. Can we put in a more mathematical basis? So we thought is, let's think about a vector. Then one thing is this is a manifold, right? Manifold for within the vicinity. So we can the tangent space. We can we can always approximate by use its neighborhood, right? So that's form a manifold. So this is Euclid subspace. So we can use that as approximate this tangent space to the manifold. So then you see this displacement vector. You can form this. You assume this form a complete, definitely not orthogonal basis, vector basis. So then you see any vector in this subspace will be linear combination of this. Okay. So using this, so you can then what arbitrary vector you can represent this. Then you got those you automatically got this projection on this subspace, right? So so this is our you can formulate. So this is we use this discrete graph representation. So therefore, if you have that in mind, then you can realize the original correlation kernel those people use can be cast into this formalism, right? In this graph representation of this vector. But the problem with those the original ones, they you see normalized. So this is really lose all the information about this how they only look at the direction, but not the magnitude of this velocity vector. So how can we add back this velocity magnitude? So we define a loss function like this. You see, well, this one is the projection, right? You know what's the best projection? You minimize the distance, right? That's mathematically give you the best projection. And this is the you still need to regularization. And this one's you see we have a B factor. This is the original correlation matrix, correlation kernel. So it means that if you we use this kind of guide because we notice the original one gives us pretty good about the direction, but doesn't completely lose the information about the magnitude. So we have used a term help us to see what the weight of this. So if your B is very big, you recover those original kernel. If you let B equal 0, then you go back to this. Okay, so this is a loss function we use to help us to determine. We can see this is really keen, right? Physics informed machine learning help determine this matrix. So with that, now the next thing is again, let's talk more about this graph vector field. So in this graph, those are data points with your data pair to represent this vector field. So we got discrete vector field. So you can there do some operation like a derivative, other things you can define right on this graph by those displacement vectors. Okay, so then you see now we talk about the full dynamics. You have given by convective diffusion equation, and then you can express the continuum PDE on a graph by this discrete representation. So or this discrete form. Then you will able now you see you essentially construct a kind of a transition, right? Markovian transition model, right? You have so each discrete point you have transition probability of the thing was well defined from your formalism. So this this is going to show you one result. With that one, you can simulate your dynamics. So this is here's a discrete simulation. So you see start from here, it evolves, and what I'm showing here is actually in the model, there's a parameter that's an adjustable parameter. It's a diffusion coefficient because what I show here in this data, my student he just used as an adjustable parameter. So he explained how this diffusion coefficient affect my result. So what I want to show the message here is this. So let me explain. This is the simulation result. A network represent simulating a differentiation process. So this tau is the overall time scale. So what he want to show here is, if you use this original kernel, you completely lose the real absolute dynamic information. So he used this time scale, right? So tau equal to 1 means that this overall dynamics is slower. This is faster. So it means you see this is we really can reflect, get this the temporal the true dynamic information, and this even this control parameter, right, adjustable parameter. You see if you adjust it, then the overall order, right, fast is fast, slow is slow, won't change. But if you want really, constrain this parameter, I think one thing one can do, he had done is in the measurement, you also have another it's the distribution. Especially you may have time series of the distribution. You can then define, to formulate another machine learning problem, try to define, try to constrain this diffusion coefficient. Then you got essentially a model without any adjustable parameter. But hadn't done that yet. But for this one, you see, we will be able to show, okay, the process, right? It's the one appear first or slower. This overall trend, the order. You agree with the experimental, the simulated result. So it means, but if you go to this kernel, then they lose all the information. They could not get this. So that's the point. I think that's good time for me to wrap up. So I want to, So here today, I'm going to discuss a lot of data, in biology, a lot of single cell genomic data, a lot of even lifestyle imaging data. Currently mainly done, those informatics type of analysis. But now we cast into this dynamical systems formalism. So we will be able to get this a lot of quantitative information, and the goal is very aligned to the goal here in this group, right? So from the data, we're going to reconstruct the governing dynamic equations. Then from dynamic equation, we'll be able to kind of predict how the system evolved and also how systems respond to different perturbations. And this is for cellular. We're talking about the whole cell. It's whole genome-wide information. So this is still very early stage, but I always want to talk to my people, right? Think about this. This is very analogous to people have done on the molecules. Proteins were big molecules. People done this half century ago. They started, those researchers started, so they can look at all this they called force field, right? Look at how the different residues or amino acids or the protein, the atom, they interact with each other. They try to find out what's the best force field. Then run simulations. They can do a lot of things. I think that we are just starting a similar adventure, but at cellular level. So with this, you see, I want to already highlight people Yan and Xiaojie who really did work, and also collaborate with my lab, Jonathan's lab, my colleague Yvette, and also VG and Eric Lander. Their people help us to generate some data, and for other, which I did lifestyle imaging, I have a lot of collaborations. So funding. I think I'll just stop here for want to hear questions and comments.
Jianhua, how many different cells have you done? I saw 3 types: neurons and I was wondering if this metrics say has the same dynamics, or if there is you can do transfer learning from one cell type to another.
Transfer learning, another thing I'm trying to, we haven't done. We now is for each data set we run this. Well, this transfer learning, in other words, it's the transferability of the parameters. That's a big issue, even for multi-dynamic simulation, because there are many other factors kind of renormalize, right, to affect this what's shown up in this transcriptome level. So how transferable? That's a good question. And then right, so that's big. We did apply to quite a number of data sets for mouse, human, zebra, stem cell, other cells, other things. Yeah, so I'm shown here.
But you didn't see if you didn't see any similarities between the A of Z, the metrics now at the the macroscopic model looks very different from one case to the other.
We haven't done those comparisons, but from one thing I'm really interested in, along what you mentioned is a cell, right? You have a cell cycle rotating, right? So those are different cells. You can define certain collective mode. Are they how are they both conserved? I think that's a very interesting question. We haven't done yet.
Yeah, so there's data I remember from Laffenberger's models where he introduced the separatrix. I think for the apoptosis model for red blood cells, long time ago, and of course there's a red blood cells at young, old, then there is like sickle cells, red blood cells in sickle cell disease, or malaria infected cells. So it would be interesting actually for these cases to see, because the underlying dynamics is the same, it's just that that there's mutations, like in the case of sickle cell, right? In the B.
Yes.
The mutation. So these are not so different, but yet the dynamics may be different. I don't know. I mean, I was just thinking loud that how one could leverage transfer learning, basically. Like
that's this is a very nice point. I have well, I didn't put it in my slide. It's very about aging, right? My student Sophia actually, she has a project that's collaborating with my colleague who moved to Harvard recently. So it's on this muscle stem cell. We want to look at the aged and the young muscle stem cell, how they respond to the mechanical stress, the mechanical substrates. And Sophia found out real indeed that their dynamics, their expression look very similar, but the dynamics, the expression, the velocity, they see dramatic difference. So there we try to infer what's the biologically, what does mean, right? Other things there. So, and also for the mutation thing, then also we can do in silico mutation, by the way, because we have
right. That's what I thought. Yes, for sickle cell is basically Heli is listening, and also Lulu. They're both familiar with this type of cells. So I was wondering if one could use your framework, maybe with neural networks to look at the transfer learning question.
Yeah, the neural network, right? So see, I think that's right. This is and also I only use here. I use this reproducing kernel Hilbert space things. Wow, you may also consider how about a neural network, right? And it's, all the things you need to find some basis, try to help as a basic function to describe the certain function, right? So that's
right.
We know, right? The neural networks give you as the uniform representation of functions. So that's something one can explore. And I found I didn't have the slide for the in silico mutations, which we can do. We show that.
So there are at least two questions that I can see.
Yeah. I see Nicholas and Tingwei want to ask questions, so maybe.
Sure. Yeah. Hello, I'm Nicholas. I'm a PhD student from Johns Hopkins University. Thank you for your presentation. My question is, what might be a good latent space if you had comparison between latent spaces to learn dynamics? I saw in some of your slides that you had UMAP. I was wondering if you had comparison between latent space of autoencoders from, I don't know, PCA, fusion maps or other techniques.
Yes, that's a very nice question. I don't like UMAP. I think UMAP is not designed for dynamics, right? If you look at the UMAP, the algorithm, you see something. It's there's some the transitions that not the mapping is not continuous. So I definitely think there's something wrong. Then yeah, what's the best way to there? That's a good question. What's the later? So that's another question for machine learning. We know there are a lot of efforts. People try to learn, use machine learning to have a neural network to find what's the best variable. So I mentioned it's the Z, right? What's the best variable? So that's one thing one can work on, which we haven't. We just too many ideas, and you need people who have that background. That's some limitation because I did mention some of the idea in this recent review I wrote. So in this field you talk about latent space, and people talk about like DMD. We may heard, right? Dynamic mode decomposition, Koopman operator. Then use there's what Cindy, right? Cindy, there are a lot of ideas how one can learn this. All deserve to try, right? See, explore. Just a lot of exciting things one can do.
Great, thank you so much. Thank you.
Hello, this is Ting Wei from UCLA. Can I ask you a question? Thank you for this talk. It's very interesting to me because it's a new field to me. And but I didn't really follow the second part. You know, you have a manifold and then you have a tangent space, you do the projection. Is your state space a manifold?
Yeah, so the state space is in all this, right? The data, right? Suppose we do the measurement, the data will form a manifold. We assume that the manifold is a good approximation of the true dynamic manifold. And then here's what I said is a pin, right? It's a, you know, we know dynamics require this should be the tangent space. You want to impose that requirement into our learning.
Yeah. So you want to move on the manifold.
Yeah.
But the manifold itself
is a dynamic system is a mapping, right? It's a mapping along this manifold.
But you want the manifold itself is fixed.
Let me see what I say. You know, so you know, for this dynamic equation, mathematics mapping from, you know, from say x time 0, you have a distribution down, then time t it evolved to another, right? And overall the data, so we this dynamic system, right? They will form a manifold. So the mapping is always within this manifold, right? The mapping from point on this manifold to the point on this manifold. Right?
I see. Yeah. So,
but then because the dynamics, so your this one, the ITT, your velocity, we know from here it should be in the tangent space.
Yeah.
So we need to impose this requirement.
Okay. So can you go to the loss function?
Yeah.
Okay. So phi I is the thing that you want to learn, right?
Yes.
And what's the relation? Is this V2 the related to phi I?
Yeah, I think.
Oh, right. All right.
Let me see this. So this is the one I saw this is people have used before, but there's certain problem, right? So here, this V, right? This is V. It should be given by, I should write down that V should be our learned phi ij then delta ij, right? The sum of that. Right?
I saw.
I forgot. Oh, wait a minute. Here,
it's not the first one. Sorry, I saw it. Okay, so you know delta ij, and you want to learn the coefficient.
Right.
Okay, and then vi is your data, right?
Vi is our data. Yes.
And
originally, right. So you know, this mathematically, you want the projection means you want this minima, right? So that's
yeah. And V2 the core is
so this is something you know, we thought originally okay, that would be enough, you know, this is regularization, this would be enough, but somehow we got not good well, because so what we'll tell the correlate is that the original people have been used as, you know, they say okay, let's just use this, because you know, only using the direction information as I mentioned, right? So if your velocity direction is along this displacement vector delta ij, so most likely, you know, they will transit from here to here, right? So that's a good the dynamics will bring you from here to here. Otherwise, you know, it'll be less likely. So that's to define a transition matrix. So we found that direction, they got direction, something pretty good. So we want to keep this term here. So depends on how much influence you have. And because my students told me if they just use this, they have trouble to get certain. My suspicion, you know, I need them to retest. A suspicion is somehow this loss function is have multiple, you know, some metastable states. So yeah, so you want maybe this one help the system to guide them to the one reasonable ones.
I see. And what is the function that people usually use for COR?
This one. This COR just the cosine.
Cosine.
Yeah,
okay.
between two vectors. Right.
I see. Okay. Well, this PIJ just reminds me of the softmax function, activation function in the neural networks. So I don't know whether they're related, whether you can design a neural network using attention and to get this PIJ and to learn it. I'm not sure about this. So can I ask a more abstract problem? Because I think for this one, it's like you learn the dynamics from data.
Yeah.
Right?
Yeah.
And I think you mentioned in some previous slide that after you learn the dynamics from data, you want to infer which path is the best path.
Yeah.
But yeah, is it like a control problem? You have
Yes, right. So I show here, so this is essentially the mathematically, right? In view, this is you can define this uses Wenzel-Friedlin formula, right? See this, can define the action. So you essentially your formula is passing. You have lot of paths, and so you try to find which one has the minimum action. So that's you cannot do variation. You find the path.
Okay. Thank you. That's very interesting. Thanks.
Okay, do we have more questions for Professor Xing? All right, thank you, Professor Xing, for your wonderful presentation.
Thank you.
Okay, so now we move to our second talk. Our second talk is given by James Basir, who also comes from the University of Pittsburgh. James is currently a PhD candidate in the Mechanical Engineering and Material Science department. He holds a dual Bachelor of Science degree in Aerospace and Civil Engineering from the Middle East Technical University. His research interests include deep learning, scientific machine learning, optimization, high-performance computing, and computational fluid dynamics. So without further ado, let's welcome our second speaker. Hi, James.
Hi, hello, everyone. Thank you for having me.
So I think you are able to share your screen now.
Sure.
Yeah.
So, can anyone confirm that they see my screen?
Yeah, could you put it in full screen mode?
Yeah.
Okay, great. Thank you.
Thanks. So, good afternoon, everybody. Happy to be here. Thank you for having me. So as you can see from the title of my talk, I will talk about neural networks that are trained on physics and equality constraints. And as you know, a lot of transport phenomena in thermal and fluid science are governed by nonlinear equations. And these nonlinear equations are partial differential equations that are very challenging to solve. And so what people do is they create these meshes or finite difference meshes and then solve these equations on discretized nodes. But there are some challenges with this approach. First of all, mesh generation could be very tedious and time consuming. There are companies that are specialized in generating quality meshes. And the other issue is that your solution can only be obtained at specified nodes. And if you have moving boundaries or problems that are changing in time, then you need to regrid, and that's adding to the complexity of the problem. And for a data-driven problem, these conventional numerical methods are inefficient and not well suited. So an alternative approach is to use artificial neural networks. So artificial neural networks are universal approximators that they're shown to be able to represent any continuous and bounded functions. And they can be viewed as mesh-free methods. So the cool thing about artificial neural networks is that once you train them, then you can obtain the solution at any point in your domain. And they're very efficient when it comes to data-driven problems where you want to embed available data to solve some problem that you have. And this is a historical perspective on machine learning that I don't want to delve into. I'm sorry, I feel like I have a different presentation here, not the updated one. I'll quickly share the other one. Sorry about that. Let me stop sharing here. This is not the presentation, the most recent one.
Yeah, it's all right. Take your time.
Yeah, here we are. This is the one. Sorry about that.
No problem.
All right. Yeah. So so here I want to talk about different paradigms of machine learning because I want to explain what paradigm we're focused on. One popular learning paradigm in machine learning is a supervised one where you have input data and labels. And so the idea is that you want to find a mapping function that maps your input to the output. And this is called supervised learning. So the reason they're called supervised learning is that these labeled data usually comes from a supervised teacher or labeler, whatever you name it. An unsupervised one would be where you don't have these labeled data. And semi-supervised is a hybrid of the two. And another learning paradigm that's pretty popular in machine learning is called reinforcement learning. And here the idea is that you want to learn an optimal behavior by interacting in a dynamic environment. So as you interact with this environment, you're reinforcing your knowledge about the environment by learning some optimal policy. And here I want to present a high-level overview of neural networks. They're nothing but parametric functions. And so here I show a very simple toy network where you have an input X and the output is U hat, and you have two parameters. For the sake of simplicity, I removed the biases. So you only have weights that I show here as theta and omega. And so what you do is you give this model input X, and this network is nothing but a series of arithmetic operations. So you see like X is fed into the input and you multiply it with the parameter theta, you get an auxiliary variable A, and then you send it through a nonlinear activation function, which is F here, and then all the way until you get the prediction from the model, which is U hat. But now you want the output of this network to be as close to the real output, which is U in this case. And for that, you need to have some sort of loss function, an objective function, and you want to minimize that objective function by tweaking these parameters. And here, like a very popular loss function here is the squared loss function that's shown in calligraphic L. And because this is nonlinear, you can't directly solve these weights for these weights. And so what people do is they calculate or approximate the gradient of these parameters theta and omega with respect to the loss function, and they use gradient descent. And this algorithm that this is called backward pass, and there are packages available that efficiently do this and dynamically, and they kind of like make use of the computed gradients and to efficiently calculate these back propagated gradients, and so that you can use the gradient descent for that. So the whole idea is that you have a loss function, and you're getting the gradient of this the theta with respect to this loss function, and then you're iteratively solving or updating your theta. So that's the whole thing with neural networks. And so now I show here supervised learning. And so for supervised learning, we assume you have some kind of data within your domain, which is shown as the capital omega, and the boundary, which is partial omega. And so I've deliberately separated the loss function for this problem into two parts. And the reason for that is that we want to study or compare our physics model that I'll later on show, because this is supervised regression models are easier to understand, easier to analyze. And then we want to have this as a baseline model and compare it against our physics model, because the only thing we would do later on is change the domain loss. And here you see that for the boundary, we have a sum of squared loss, and for the domain loss, the same thing, because we assume we have data for both the boundary and the domain. And one thing that I should point out here is that these two losses have the same scale. And later on, what we do is we say, well, what if we do not have data for the domain, and you have some function, some operator, some differential equation, whatever you name it, that I showed here as a calligraphic R. And you say, well, my output within the domain has to satisfy that. And so what do? We just replace that the loss for the domain with that equation shown as calligraphic R. Now, these two could be easily compared, but one thing I should point out here is that now the loss function for the domain and the boundary, they don't share the same scale, because one of them has derivative operators and physics. The boundary has maybe Dirichlet, Neumann, could be anything, but they don't share the same scale now. And by comparing these two models, now we can understand what's happening when we embed physics in the model, how the loss landscapes change, how the model learns, or what problems we face and how we can tackle those issues. And I want to get into the history of physics in neural networks a little bit. This paper is by Dissanayaki and colleagues. It was published in 1994. And so what they do is they say, well, if you give me a physics operator that's calligraphic L and a boundary operator that's calligraphic B, I could write two loss functions and combine them together into an objective function and solve it or train my network on it. And hopefully the predictions of the network would give me the exact or the correct solution. And they've shown it on some simple Poisson equation, but at the time people didn't know about neural networks, and there were no PyTorches and TensorFlows and all of these packages available. And so they didn't have the flexibility to kind of like create different architectures and see what happens. And so they manually would approximate those gradients and kind of kept their architecture fixed and trained the whole thing on that. And another paper that was published in 2003. So the authors here, what they did was they were like, well, instead of taking the sum of squared errors on the domain and in the domain and on the boundary, let's take the mean of the two. And so they've shown it on some problems that, but again, it was 2003 and people didn't know a lot about neural networks. And until 2012, which the ImageNet thing happened, and people knew about neural network, and then people kind of like adopted the method and appreciated it. Different from the previous authors, in 1998, Lagares and colleagues proposed a different one. So their idea was like, hey, instead of having these two objective functions, we want to create a trial function for our operator, for our problem, that this trial function is satisfying the boundary conditions automatically. Now you only have one objective because the trial function is satisfying the boundary, so no need for the boundary loss. This is a really nice idea, but it has its own issues. Well, first of all, it's really hard to create trial functions for a high dimensional problem, where if you have the boundary points available at a sparse location, then it's just a tedious thing to write a large trial function. Another issue, big issue in my opinion, is that it's hard to know which trial function is the best one. You know, and the whole point of using neural network is not to impose our own bias on what the solution of the thing should look like. So we want the model to learn it. Anyway, here I show it on a very simple one-dimensional Poisson equation with homogeneous boundary condition. You can see that I can write infinitely many different trial function that satisfies the boundary condition automatically. But there's no way of knowing which one of these solutions are optimal. And it's possible that I pick a really bad one and try to learn that, but it's possible that it doesn't work. And so that's one of the issues with these methods. That's why they're not really used a lot. And the modern implementation of physics-informed neural networks, in my opinion, the paper, the key paper that really helped neural networks get acceptance or appreciation in the scientific community is the one by Raisi and colleagues. And it's the same thing, but they implemented on with modern deep learning frameworks, and they solved some really cool problems on that with these methods. And so the idea is, again, the same. You have physics and you lump these two losses together. But like I said, now these two objective functions have different scales. And from an optimization point of view, combining loss functions with different scales into a single objective function is not favorable, is not, I should say, suggested to do, because it's like apples and oranges lumped together and you're trying to find an optimal thing. And we showed this by comparing these models with baseline regression model on a one-dimensional Poisson equation, and we're like, what happens if we increase the complexity of these problems, and why pin models fail? So here I show a sinusoidal one-dimensional Poisson equation, and the reason I use a sinusoidal one is because I could increase the complexity of the solution easily. And you see that when omega is 5, a pin model is producing 2 orders of magnitude better solution than a purely regression model. But as we increase the complexity of the solution, pin model fails. However, regression model keeps almost similar orders of magnitude error levels. And we wanted to know why that happens. But we do know that, okay, for the regression model, these two objective functions, they have the same scale. But for physics model, all we did was put physics instead of the domain loss.
Hey, Shane, sorry, can I interrupt for a minute?
Sure.
Yeah, could you go back to the previous slide? So here, what do you mean by regression model? So for regression model, what information do you have? Do you have observations?
Yeah, so for the regression model, instead of physics, we generate points from the exact solution because we know the exact solution. So we train it on data, basically.
Okay, but for pin, you don't have data, you have residue.
Yeah, you only have the boundary condition, but now instead of exact data within the domain, you use those points to basically calculate or approximate your physics loss on the physics operator, which is du squared u dx squared.
Yeah, okay. And another question is, like, what is the architecture of the neural network you use for those two tasks?
Oh, it's the same thing. The architecture is like a feed-forward neural network. I believe it was a 8 hidden layer network. You could check out our paper, like we explained.
Okay, great. Thank you.
There, yeah. But everything is the same, like the optimizer, the learning rate, architecture, everything is the same, except for the purely regression model, we have exact labels, but for pin, we don't.
Okay, got it. Thank you.
And then, sure. And then we looked at their loss landscapes to see why that happens, or whether we could get something from looking at the loss landscape. Turns out that purely data-driven network that's trained in a supervised fashion, where you have data, kind of gives you this nice landscape where the model is able to find a good local minima. But for pin model, when we increase the complexity of the solution from omega 5 to 10 to 15, the loss landscapes becomes chaotic and very hard to optimize. And so, and we know that the only thing that we changed in our loss function was embedding physics instead of data, and that was it. And what that did was created a scale disparity. And people then realized that, well, now how could we fix that scale disparity? They were like, well, let's multiply these two objective functions with different lambdas or different weights. Now that's a really cool idea. That helps with removing that scale disparity. But now the question is, what are those weights? In conventional machine learning, you have some called validation data set where you use that data set to tune your hyperparameters. But for solving a PDE, you don't have data. And so there's no way of knowing what are these weights. And there are a couple of published methods that I'd like to talk about. One is the one proposed by Wang and colleagues. And so they propose something called empirical learning rate annealing. And so they say, well, if you have an objective function that's given in equation 1, how do you find, or how do you approximate those lambdas? And so they said, well, let's match the backpropagated gradients, and then based on that, find these lambdas. But the problem with this is that, you know, from high school calculus, we know that if you want to find an extreme point on a function, what you do is you equate the gradient of that thing to 0. And so you want the gradient of whatever thing, the extreme locations on function, to be equals to 0. And so in optimization, that's called optimality condition. And so you want the optimality condition to be satisfied. But by updating these lambdas that's given in equation 3 the way it is, equation 2 is not necessarily satisfied. And so what happens is that once your training is done, you land somewhere in the space of the parameters and you think that that's a local minima, but it's not, because mathematically you're not satisfying the optimality condition. And another issue would be like, if you look at equation 3b, when you're converging to a local minima, your losses get smaller and smaller. And so theoretically, when Li approaches 0, your lambda hat I blows up. And that means you have a very large learning rate. And that could that could make the model diverge or miss on the local minimum. So another approach that was published by Vandermeer and colleagues, they were like, well, let's take the affine combination of the two. And so what is a affine combination? So a affine combination is like finding these two weights in a way that they're between 0 and 1, but their sum has to be 1. And that means that you're favoring one loss over the other. Now, this is also a nice idea, but because these two objective functions doesn't share the same scale, finding these two weights doesn't necessarily tell you which objective is favored relative to which objective, because they don't share the same scale. And so we thought, well, how about, and so here, let's formulate the problem first, and then we'll move on to how we tackle this problem. So we assume we have some noisy measurement data, a set of like xi and u tilde I. So we have m of those observations, and we have a PDE, a governing differential equation that is governing our problem. And we also have our boundary operators. Now here I assume that your boundary conditions are exactly known. And in case you do not know your boundary conditions, you could quickly say, well, now my boundary conditions could be treated as a noisy measurement data. And so the idea here is that we want to find a mapping function that integrates our measurement data and also obeys the governing equation, as well as satisfying the boundary condition. So that's the whole objective. Well, for the noisy measurement data, it's very easy. We just use a likelihood estimation. So we assume that the prediction of the neural network with some noise would give you the measurement data. And so with that, we assume that the errors of this whole observation is normally distributed with mean 0 and a standard deviation A. And this is just basically minimizing a log likelihood, and you get this objective function, which is calligraphic J sub m theta. And then we move on to creating an objective for our governing equation, which is, I believe we all know, it's just a simple, you put the collocation points in your PDE, and you sum the square of all of these residuals, and that's your physics loss. Then we say, well, now we have an optimization problem here, that we want to minimize our physics loss with our noisy measurement data, such that our boundary losses are within some interval minus epsilon to epsilon. And why we do that is because, like, it's okay if you have minus 10 to the whatever minus 3 or plus 10 to the minus 3 error in that range, because we want this thing to be in a very small error range. And now you could easily see that, well, hey, instead of minus epsilon to plus epsilon, why not take the absolute value of the thing, right? And you could. And so without loss of generality, we said, well, now we could that could be a phi function. And so that phi function could be an absolute value function, a quadratic function, anything, a convex function that's between 0 and infinity. And why it's positive? Well, because we care about the distance of error, not about the sign of it, right? And now you could easily push epsilon to 0 and get an equality constraint here, like I show here, phi of beta for all x equals 0. And now you have your physics and equality constraint optimization formulation. And you could use any unconstrained optimization formulation like penalty method or Lagrange multiplier method or augmented Lagrangian method to convert this problem back to an unconstrained loss function where you can use it to train your model. And so here I use augmented Lagrangian method because it's basically in a way combining the Lagrange multiplier method and penalty method. And that's shown in equation 4, because if you get rid of penalty, you get the Lagrange multiplier method. If you get rid of Lagrange Lagrange multipliers, you get the penalty method. So it's kind of combining the benefits of the two. Now, here we use, I used augmented Lagrangian method to convert our constraint optimization problem back to an unconstrained optimization problem. Now, these, because of the augmented Lagrangian method, you can iteratively update your Lagrange multipliers. Now you don't have any scale issue, no problem. And you make sure, and because we properly formulated it, now we make sure that after the optimization is done, our constraints are strictly satisfied. And I show here, like, kind of like, we discussed this in our paper, like we show like why absolute quadratic function is better than absolute value. But here I want to give you a little math here, not really complicated, but it's just a simple math where you see, well, let's say instead of like, let's say phi is an absolute value function, right? And so if you take the gradient of this, which is case one, what you do is the gradient of that with respect to the parameters of the network, which is theta, is basically lambda gradient of u hat. And so now at that time of training, the current state, the error, which is u hat theta minus u, is lost. Like you don't have that information available no longer. But if you, instead of like absolute value or a hat function or unit function, have a quadratic function, now at each step of optimization, your parameters are getting updated and they have the information of feedback on how much error you make on your boundary loss. And these are some of the metrics that we have here to evaluate these models and methods. And now for the same problem, same architecture, same optimizer and hyperparameters and everything, we apply this method to see what happens. And it turns out that this way of formulating an objective function is not only producing better results for simple problem, but it sort of keeps that level of accuracy for very challenging solutions as well. And the details of this whole thing is in our paper that we published in the AAA SciTech. And we look at the loss landscapes here. It turns out that the Pecan way of formulating an objective function kind of have better loss surfaces that the optimizer was able to converge to. And then here we show that, well, you could apply it to very like irregular domains as well, because I mean the objective function is not necessarily domain dependent. You don't have to have a regular domain to apply these methods. And this is another Helmholtz benchmark problem where we solved, and you see that we only use 512 number of points on the domain in the domain and 64 points on the boundaries, and we're able to get like 2 levels of accuracy, better solution than our baseline method. And now here we show that, well, you could do this, you could use this method to do multi-fidelity data assimilation as well. And figure A shows that we have two different fidelity levels of 2 different data here, noisy and clean data or high fidelity data. And this is just, so the idea here is that you want to learn KH, which is the soil conductivity, but you're given pressure hit, which is H in this case. And so we know, just to tell you like what these high fidelity and low fidelity pressure hit corresponds to the soil conductivity, it turns from figure B, you see that the low fidelity is way off than the exact solution. But we don't want to like get rid of these low fidelity data. We somehow want to embed it in our model, and we want the model to extract these information from low fidelity data. Turns out that our model is able to accurately predict H and K with high fidelity data alone. But if you add low fidelity data in your training as a noisy measurement data, which makes your model a multi-fidelity model, then you get better predictions with way better error levels. And this is another benchmark problem that was discussed in Van der Meer and colleagues. And so here we use like uniform sampling. not adaptive or anything, and we use the same architecture and the same number of points. Turns out that when your phi is a quadratic function, you get one level of accuracy better than their method. And when it's Huber, you get 2 orders, I believe. I can't see this thing here. Yeah. And this is another benchmark method that was recently studied by somehow, I don't know, my Zoom task thing is stuck on this. Yeah. Okay. So this is a purely convection equation. And so here U could be any physical quantity that's convected or transported with a velocity b. And so b in this case is 40. And we want to solve this equation with our method. And so this was, so the authors in this paper, Aditi and colleagues, proposed what's known as curriculum learning. And so curriculum learning in conventional machine learning. So the whole idea of curriculum learning is that you have simpler set of data, and then you gradually increase the complexity of your data, and you allow your model to gradually learn this whole thing. So the challenge is that how do you sort this whole data gradually for conventional machine learning? But in our case, like, yeah, for this one, I know that if I increase beta from 1 to 40, the model will gradually learn that the solution get complex and complex. But for any like given PDE, I don't think there's any way of knowing like what should I change in this PDE to get an easier solution that makes it easier for the model to learn and then increasingly make it complex. And the fact that you start with a simple model, how many times do you have to train this? And so it's kind of computationally expensive. But what we do is we just solve it with beta 40. Let's formulate this thing and solve it with beta 40. Turns out that if you set your phi quadratic function, you get 1 order of magnitude better solution than the baseline method. When it's a Huber, you get 2 orders of magnitude better. And you're only training your model once. I believe, okay. So I believe with that, I've come to the end of my talk, and here's the link to our published paper, and also the link to our archive version and our GitHub page, and also my own GitHub page where I have a lot of problems and projects we do. And now I welcome any questions you might have.
Thank you, James.
Thanks.
Do we have any question for our speaker? Let's see. Hey, James, do you have any follow-up works? So this is the most recent work, right? You're talking about in this talk.
And not really. We actually have a follow-up work that hopefully I'll be able to share and publish the codes and everything, but I didn't put those results here yet. But we do have some really, I should say, interesting work that's coming up pretty soon.
But they're not on like your GitHub website.
Not yet, but I'm planning to, like, these days, maybe in a couple of weeks.
Oh.
My advisor is going over the manuscript and everything.
Oh,
So,
okay.
yeah, everything is done. I'll post it on GitHub and everything.
Yeah,
But
yeah.
I'd love to have comments on the GitHub if people have like developed it with PyTorch, and you could clone it and try your own models.
Sure.
you
Yeah.
know.
Yeah, we'll take a look at it once you release your results and codes.
Sure.
Okay, since there's no question for our speaker, thank you, James, for your wonderful presentation. And yeah, and this is it for this week's Crunch seminar. Everyone has a good weekend, and see you next week.
Thank you. Have a great day.
Okay, bye.